Chromosomes, the blueprint of life. Chromosomes are structures made of DNA and protein located inside the nucleus and responsible for storing and transmitting genetic information. The function is to help to ensure that genes are acquired, copied, and distributed during cell division. Humans have 23 pairs chromosomes, and maintaining this exact number is essential for healthy development. The example is Down syndrome. Down syndrome occurs when there are 3 copies of chromosomes 21 instead of 2. This causes symptoms such as developmental delays and cognitive impairments.
The second is about the relationship between DNA, genes, and chromosome. Before explaining the relationship, let's look at the definition. First, DNA is a double helix structured molecule that contains genetic information and controls all biological activities. Next, genes are specific parts of DNA that serve as units of genetic information, and it determines proteins and various bodily functions. Lastly, chromosome is a structure made of DNA and histone proteins, which organize and transmit genetic material during cell division. The relation of these three things can be like genes constitute DNA and DNA makes chromosomes. The real world example can be like the genes that determines skin color is located in a specific region of DNA, and this DNA is found in chromosome 16. We could understand these relationships by giving an analogy that chromosomes can be bookshelf and DNA can be a book inside that bookshelf, and the genes can be one chapter or page inside that book.
This part is about human chromosomes and karyotype. First, human cells contain a total of 46 chromosomes in the nucleus. 22 pairs are autosomes and 1 pair is sex chromosomes. Each parent contributes 23 chromosomes to their child. Next, a karyotype is the number and visual appearance of the chromosomes in the cell nuclei of an organism or species. Karyotypes can be used for medical examinations and to diagnose genetic disorders such as Down syndrome or Turner syndrome.
In cell division, chromosomes are responsible for the accurate transmission of genetic information. Mitosis divides somatic cells into two genetically identical daughter cells, during which sister chromatids are separated. In contrast, meiosis produces gametes with homologous chromosomes separating during meiosis I and sister chromatids separating during meiosis II. Genetic variation is ensured through crossing over during meiosis. However, errors such as non-disjunction can result in gametes with N+1 or N-1 chromosomes, potentially leading to genetic disorders like Down syndrome. In mitosis, abnormalities in the spindle apparatus can cause uncontrolled cell division, contributing to the development of cancer. Thus, understanding chromosome segregation mechanisms is essential for the treatment of genetic disorders and cancer research.
In this last slide, I will explain how chromosome technology impacts
model search. First, genome mapping and tools like CRISPR-Cas9 allow scientists to edit genes with precision. This has huge potential for treating genetic diseases. Second, chromosomal analysis helps in medical diagnostics. For example, detecting cancer-related abnormalities or predicting genetic disorders during pregnancy. We also see advances in personalized medicine, where treatments are tailored based on a person's genetic profile. Lastly, in agriculture and biotechnology, cross-chromosome technology is used to improve crops and research animal genetics for better productivity. In short, chromosome research is a powerful tool shaping the future of health, science, and agriculture. Thank you.